Assalamualaikum and a very good day to everyone. We are from Group 5 and the first presenter is Muhammad ZAKWAN, which is me that going to present about the trace member protein. After that will be the second presenter, which is Shazly Iman, that will present about the peripheral protein, and lastly the last presenter is NURSYAMIMI that will present about the enzyme. Okay, so let's start with the trace membrane protein. So the trace membrane protein that I choose is the ER membrane protein complex subunit 6, which is EMC6. EMC6 is a part of the endoplasmic reticulum membrane protein complex that enable the energy independent insertion into endoplasmic reticulum membrane of newly synthesized membrane protein. This protein is also involved in the translational insertion of multipass membrane protein in which stop transfer membrane and consequence become ER membrane spanning helices. The endoplasmic reticulum is a network of membrane-bound structure that also play a key role for the protein synthesis and folding. This function of the endoplasmic reticulum can lead into a wide range of disease, including neurodegenerative disorders such as Alzheimer's disease, as well as other conditions like diabetes and cancer. Okay, so for the FASTA sequence, we get from the UniProt, which is here for the EMC6. This is the FASTA sequence of the EMC6. And for the binding, okay, next for the domain and binding site, EMC6 has four topological domains, which are cytoplasmic and luminal, and four transmembrane, which are helical. Okay, next to the structure of the protein, which we get from the PDB of the EMC6 is the 6WW7 of the structure of the human ER membrane protein complex in a lipid nanodisk. So we can see it in the sequence, which is they have so many chains, and for the ER membrane protein complex subunit 6 is on the chain F. Then we transfer into the UCSF chimera. This is the full structure of the protein, and after that we can see that this each color that represent to the every chain of inside the protein, and for the chain F is labeled on the red color for the EMC6 and also in a ribbon structure. And next to the secondary structure, we can see for the alpha helix is labeled on the blue color, beta pleated sheet is in the green color, and the coil is in red. For super secondary structure for the EMC6, we can see for the subunit for the beta barrel, we can see the form of beta barrel at here, and also the beta Mendel pattern. Next to the mutation, the mutation sequence has no effect for any kind of disease, but it can decrease membrane insertion of hydrophobic transmembrane helix containing protein by the EMC. These are the two sequences that already been altered, which is from the sequence 27 is D2A, which is aspartic acid, to alanine, and sequence 31 C2A, which is threonine to alanine. That's all from me. Thank you.
Assalamualaikum, my name is Shazly bin FANSURY, and today I'm going to present to you my part, which is peripheral proteins for this group. So what is peripheral proteins? Peripheral is also known as peripheral membrane proteins. It's a group of biological active molecules that are composed of amino acids interact with the surface of cell membrane lipid bilayers. So peripheral protein does not enter hydrophobic area. So the research that I did for peripheral proteins are the cytochrome C, that cytochrome C play a crucial role in process of cellular respiration and also in electron transport chain. So the protein is a small water soluble, contain protein that transfer electrons between complex 3 and complex 4. So I'm going to move to FASTA format. So the FASTA format that I fetched from UniProt is from UniProt. So this is the protein name, and this is the gene code. So that I fetch is sequence here, and then just click download, and it will bring you to this is the FASTA format for cytochrome protein. So as you can see, this is cytochrome FASTA format. So moving on to the next part is the domain binding site of protein. So the domain binding site of protein is there's a two topological domain, and one is transmembrane, which is helical, and the other is mitochondrial matrix and also mitochondrial membrane. So as you can see on UniProt is on uh, subcellular, if I'm not mistaken, subcellular. This is the part which is transmembrane topological domain, mitochondrial matrix, and also mitochondrial membrane. So next is I'm going to move to protein structural model. So this is based on Chimera. So Chimera. So this is the figure that I fetched from Chimera. So as you can see, there's a lot of there's a lot of chains in the protein after 3D structure. Uh, on and the figure that below shows the structures of uh, cytochrome oxide 6 subunit 6C. So as you can see, there's 14 that are obtained from PDB, and after that I entered the ID for the cytochrome, which is I can fetch it from here. So this is the PDB. This is the code that I need to fetch and then enter it into my Chimera, and I will get this figure. So this figure is after I immerse into the secondary structure. So the secondary structure is here. Flowing on to this one, as you can see, the chain that the chain that happens on the cytochrome oxide is on the white one. The white one is I, so which is the white one is the chain I colored. So moving on, next is the labeling protein. So labeling protein is this. The first one is the secondary structure. The first picture depicts the ribbon structure, and also the next one, whereas the second picture depicts the color of the secondary structure, which is blue for helix, yellow for coil, and also purple for strands. So as you can see, based on UniProt, uh, my apologies, based on Chimera. So this is the image that I get on the PDB. As you can see, the blue is helical, yellow is the coil, and also purple is the strand. And as you can see, the strand. So there's if you examine clearly, if you rotate the object, you can see there's a barrel here, which is the super secondary structure. Is a beta barrel. So in this peripheral protein cytochrome, there's a peripheral there's a beta barrel here. So moving on to the next one, which is I already explained, is like a cylindrical shape.
Okay, next I will present about pyruvate kinase 1 for enzyme. The function for pyruvate kinase 1 is to catalyze the formation of pyruvate in the last step of glycolysis. It is irreversible under psychological condition. The reaction is critical for the control of metabolic flux in the second part of glycolysis. The genetic defect of this enzyme causes the disease known as pyruvate kinase deficiency. In this condition, a lack of pyruvate kinase slows down the process of glycolysis, will affect the cell that lack of mitochondria, because the cell must use anaerobic glycolysis as their sole source of the energy because the TCA cycle is not available. For example, red blood cell which is in a state of pyruvate kinase deficiency rapidly become deficient in ATP and can undergo hemolysis. Therefore, pyruvate kinase deficiency can cause chronic uh, non-spherolytic hemolytic anemia, or the short form is CNSHA. Next, the FASTA. The FASTA is I get from the Uniprop, from sequence. From the sequence, I click the button download and it will show this FASTA. For this enzyme, it has a binding site where it have 14 binding site and one site. This binding site, the sequence is RRNNFSH and the site for sequence is K. The protein structure is I use the UCSF chimera to get the protein structure, and from this structure I color it by the different color to differentiate the chain within the enzyme, where it have 4 chain. It have chain A, B, C, D. For chain A, the color for red, and chain B for yellow, chain C is green, and the chain D is for purple. In this enzyme also it have a secondary structure where I color it by 3 color. For the helix, I color by magenta, and the strand is by color cyan, like a blue like that, and the coil is dim gray. Here, dim gray. The coil is works like to connect between secondary structure. And for mutation, it is caused by autosomal recessive trait, where the mammals have a 2 pyruvic kinase gene, PKLR and PKM, but only PKLR encodes for the red blood isozyme, which affects pyruvic kinase deficiency. Over 250 PKLR gene mutations have been identified as associated with pyruvic kinase deficiency. DNA testing has got the discovery of the location of PKLR on chromosome 1 and the development of direct gene sequencing tests to molecularly diagnose pyruvic kinase deficiency. Okay, last is about factor and method. For factor, the enzyme can happen is because temperature, pH, concentration, substrate concentration, and the presence of any inhibitor or activator can have an impact. It works best at specific temperatures and pH levels, and at the poor condition can cause an enzyme to lose its ability to bind to a substrate. The conformation or shape of a protein together with any post-translational modifications such as phosphorylation or glycylation might influence in stability. Additional factors that could impact protein stability include ionic strength, detergent presence, protein concentration, and the presence of other proteins. However, there are several methods that can be used to determine the structure of the protein. The first is X-ray crystallography, where this technique is used to determine the atomic and molecular structure of a crystal. The crystallographer will obtain protein crystal record, the diffraction pattern formed by X-ray placed into the crystal, and then use a computer to input the data. The goal of X-ray crystallography is to determine the three-dimensional molecular structure of a crystal. The second is electron microscopy, where it is a technique that produces high-resolution image of biological and non-biological specimens. In biomedical research, it is used to investigate the detailed structure of tissue, cells, organelles, and macromolecular complexes. It's also a microscope that uses an accelerated electron beam as a source of illumination. That is nuclear magnetic resonance spectroscopy, or an NMR, where it's a technique based on the interaction of radiofrequency radiation applied externally with atomic nuclear. During this interaction, there is a net exchange of energy, which causes a change in nuclear spin and intrinsic property of atomic nuclear. The properties of the atomic nuclear in the protein are used to determine the structure of the protein. The last is bioinformatic tools, where we as a student bioinformatics can create the software program to extract meaningful information for large molecular biology or biological database and to perform sequence or structure analysis. But in this each method, it has both benefits and drawbacks. The method of choosing is specified by the protein size, stability, and solubility, as well as the resources availability. I think that's all from us. Thank you for watching our presentation.